Hello, my name is Serena Duchi and I'm a cellular biologist and I'm working as a research fellow at the University of Melbourne inside the St. Vincent's Hospital. So my research work is based at the Biofab 3D, which is the first hospital-based biofabrication or 3D bioprinting laboratory in Australia, and is set inside the St. Vincent's Hospital in the heart of Melbourne. Here, researchers, engineers, scientists, clinicians, and also industry partners are working together to build biological structures such as bone, cartilage, muscles, nerves, and also other organs. So almost anything that requires a repair from a disease or physical trauma. And my research is in particularly focused on the repair or regeneration of bone and cartilage, which are the main components of the skeleton. The skeleton is the supporting framework of an organism, and it is typically made of a hard, rigid tissue that supports the form of the animal's body and protects the vulnerable organs, but it also performs many essential functions. So some are directly related to the purpose of all the skeleton, so providing structural support, protection, support for locomotion, but it is also involved in other really important functions such as blood cell production, storage, endocrine regulation. So that's why it's really important to protect the skeleton and to protect it from some of the diseases or conditions that can happen, because the skeleton, as many other organs in the human bodies, is really fragile and vulnerable. Here in these slides, I can show you some of the examples of these conditions, such as arthritis, osteoporosis, scoliosis, but even fractures and tumors can happen. So the main question is, how do we repair bone and cartilage without the need of removing them, without the need, for example, of an amputation? And to answer this question, we can go back in history to the work of Nicolas André de Beauregard, which is considered the inventor of the word orthopedics and of the relationship in the management of diseases between the happiness of the doctor and the obedience of the patient. And really famous is the drawing of the crook tree in his book Orthopedia in 1743, when it was one of the first one in introducing this pioneeristic approach of the conservative treatment. So as it can happen with a crook tree, a gradual straightening of the limb by bandaging the bent limb can spare the limb and save the limb by itself. But science and discoveries during these years have progressed, and until the discovery of this special population of stem cells, which is called mesenchymal stem or stromal cells. Those are adult stem cells, so they can be found in many tissues inside the adult human body. They are considered immature and undifferentiated. Contrary to the embryonic stem cells, which are considered totipotent, MSCs are multipotent, so meaning that they can generate connective tissue types. Since they originate from the mesoderm, where the connective tissue develops, they can originate and they can differentiate into multiple tissue types, but only muscles, bone, fat, and cartilage. They have the potential to self-replicate as the embryonic stem cells. They can fight apoptosis, so meaning they can fight the cell death, and they can reduce inflammation. And another important function that has been discovered more recently is the so-called tumor homing ability, which means that in 5 different steps that they have been identified during the years, these stem cells can recognize the signals from the tumor cells or released from the tumor cells, and they can literally migrate towards those cells and integrate some home into the tumor stroma. So that's why us and many other groups had the idea to use these cells to transport actually drugs or chemotherapeutic drugs in order to have a better targeting of the chemotherapeutic drugs inside a tumor, and for example, a bone tumor. So just to sum up, the MSC's applications are quite wide at the moment, but our research is mainly focused on cell therapies and drug delivery systems. And the 2 main focus, as I said, are cartilage repair and bone tumor treatment. So we can use these cells as a sort of Trojan horses or workhorses, rebuild the cartilage, to rebuild the sick bone, and to have a functional and healthy limb at the end. So for what it concerns the cartilage, the clinical problem that we are trying to tackle is to prevent the onset of osteoarthritis. So what can happen to a normal cartilage is that an injury, even if it's small, it can progress and deteriorate into this really chronic and painful condition, which is called osteoarthritis. And the main or the current surgical treatments also in our hospital, the St. Vincent's Hospital, are mainly two. So one is called microfracture, and it's basically a sort of conservative approach where you actually stimulate the stem cells underneath the cartilage by creating little holes inside the cartilage and the subchondral bone, and you stimulate the stem cells to actually regenerate and rebuild a healthy cartilage. But this technique has got many drawbacks and many limitations, and some of them are the creation of a sort of a scar tissue, so fibrocartilage, which at the end is even more painful than the osteoarthritis by itself, and it creates a lot of inflammation. So the second surgical treatment and the final outcome is the total joint knee replacement with the use of prosthesis. And as you can understand, this is a sort of really harsh clinical challenge. So in terms of millions of people affected by this disease or condition in Australia, we are talking about 4 million every year. And this number is due to increase in the years to come. And also in terms of economical cost to the health care system, the numbers are due to increase in 2030 and are expected to rise up to 7.6 billion. And so that's why we had the idea of try to find alternative strategies through the use of stem cells and tissue engineering to revert the osteoarthritis and to possibly cure and treat a small injury and to regenerate and repersonate a normal cartilage. So what we do as a vision, as represented here in this cartoon, is to take a tissue where we can isolate the stem cells from, which is called infrapatellar fat pad. It's a fat tissue basically located underneath the patella, which is close to the articular cartilage of the knee. Then we do isolate these stem cells and we mix them with a chondrogenic hydrogel, meaning that is a gel, which is biocompatible, and it contains small ingredients to help the stem cells to actually become a cartilage. And this combination of MSCs and chondrogenic hydrogel, we call it bioscaffold. And the way we do implant the bioscaffold inside the cartilage defect is called in situ implant. And with the help of a light, we do induce the hardening of this material and ultimately the cartilage repair. The need of having this so-called in situ implantation is that because actually and currently the imaging techniques still fail to identify small gaps of the defects, so impeding the so-called self-production of a perfectly matching bioscaffold. So to deliver at the time and at the point of need a bioscaffold capable of regenerating the deficient tissue, the so-called in situ biofabrication, it can help to provide a really nice outcome. But since we cannot, not yet build a 3D bioprinter around a human knee or human legs, our surgeons, Professor Peter Chung and the scientist Professor Gordon Wallace, had the idea a few years ago to create and to produce the so-called handheld device, handheld 3D bioprinter device, also called the Biopen. The way this device and this handheld portable 3D bioprinter is working is that it's able to extrude the stem cells mixed with the chondrogenic hydrogel, so the bioscaffold inside the cartilage defect. The bioscaffold also contains a small molecule called photoinitiator that when it's eaten by a light, it favors the hardening of this gel. And so inside we have the creation and the formation of the stem cells plus the hydrogel, so a hard material that it contains a lot of, cells, 99% viables, and that are able to load the bearing of the knee. So, because we have to remember that the cartilage is a tissue that functions to actually bear the forces, for example, when we walk. And ultimately, as I said, we obtain and we achieve the cartilage regeneration. Another feature of this handheld device is the so-called coaxial design. So the Biopen is designed in a way that we have two compartments. One is core that mostly contain the stem cells, and the other one is core shell, okay, that mostly contain the hydrogels. And here you can see in these confocal pictures that in green is the compartment where we have the cells, and all around the shell underlined in labeled in red. With this configuration, we can achieve the maximum of cell viability, and as I said, we are talking about 99%, and also the maximum of the stiffness in order to bear all the loading. And of course, ultimately, we can achieve cartilage regeneration, so neocartilage formation. As you can see from this picture, this is a confocal images of one section of the bioscaffold. In blue are the cells nuclei, in red the cytoskeleton, so the morphology of the cells, and then in green here you can see appearing the nice cartilage layer right just after 21 days in a culture. So this is what happens in vitro, and we have studied all the conditions to, improve and always have a better cartilage. But we also prove this in a preclinical animal model. These are a few images describing the workflow and what we have done using a sheep as an animal model to treat cartilage defect. The sheep is, let's say, the best model system in this case to assess the cartilage regeneration. And we are in a phase of analyzing all the data that we got out from the study. It was a 6-month study, and here is just an example of a 3D reconstructed image from MRI, so from magnetic resonance. And on your left-hand side, you can see the microfracture procedure, and you can see the layout of the actual cartilage defect, which has not been repaired, but instead has created a really strong inflammatory reaction underlined labeled in red. Whereas on the other side, on the contralateral side, we can see the effect of our treatment. So almost, let's say, 80% of the cartilage has been repaired and has been restored. During those years, during those 4 years, we have also modified the design of the Biopen, and we have now a really nice prototype. It's called MK 4.2, and we have also created our own company, which is called Axelda. So we really hope that in a few years' time, possibly 1 or 2 years, we will move towards the clinical translation. So we will activate the first clinical phase 1 and 2 clinical trials in order to assess the safety and the efficacy of our strategy in humans. In the meantime, the other project that we are working on, and I'm particularly interested in, is the treatment of bone tumors. And is in particular the treatment of the osteosarcoma. So this tumor is the most common primary tumor of the bone. It prevalently it eats people and in pediatric age, so between 10 or 25 years old people. It eats the distal metathesis of the growing long bones. It is characterized by osteoid, so a bone tumor formation that is invading the surrounding healthy tissue, and is also characterized by highly proliferative cancer cells that they absorb the healthy bone. But without going much into the details of this disease and this type of bone tumor, what is clear is that during the years, the treatment and the 5-year survival rate of the patients hasn't increased and hasn't improved much. So we are still talking about 60 or 70% 5-year survival rate. So there is a need, to find novel strategies to actually try to treat and to spare the limb. So that's why, as I said at the beginning of my presentation, the MSCs, since they have this tumor homing capability, they can be used as a Trojan horse or as an anti-cancer Trojan horse. So they can be used to internalize chemotherapeutic drugs or internal or external trigger cell death stimuli, and in a way that they can, when they migrate and they home into the tumor stroma, they can release the chemotherapeutic drugs and eliminate and kill the tumor cells, but also themselves. So it's a sort of suicide effect in order to ensure at least 90% of tumor cells removal. And in this graphical cartoon, this is what we do, and this is the vision that we have in order to apply this strategy in the future in affected patients. So the source of cells that we use in this case for this particular treatment is the bone marrow. We do, as we do for the cartilage, isolate the bone marrow-derived MSCs, and we load them with the chemotherapeutic drugs or with the so-called photoactivable drugs. So in a way that once we in situ deliver this combination, well, these stem cells loaded with the drugs inside the tumor, as we do for the cartilage repair, we can photoactivate, so with the help of a light, as we do for the cartilage, and so the drugs, and so we can kill tumor cells and the stem cells by themselves. This is a graphical representation of the in vitro two-dimensional, the so-called two-dimensional experiment that we have done in order to prove that the therapy can be efficient. And here is a video showing in red the tumor cells in a 2D surface, and here the other cells with the green dots are the cells, the mesenchymal stem cells, that they have internalized the chemotherapeutic drugs or photoactivable drugs. And in this second video, you can see that when we hit even one single MSC is loaded with the drug, we can eliminate almost 100% of the tumor cells and the MSCs by themselves. Everything is working really nice in vitro, but of course we need a proof of concept in vivo even in this case. So we ran several preclinical animal models using mice, and we use a really nice strategy through which we can monitor the tumor cells through emission of light with a system that is called luciferase. So we can always monitor where the tumor is and how it's growing and how it is eventually reduced thanks to the therapy. And on the other side, we have the MSCs loaded with the drugs, and those drugs that can be labeled, so we can always know where the drugs, so the MSCs are, and if they can contribute to the elimination of the tumor. These are the results, one of the most amazing results that we obtain by inducing a subcutaneous osteosarcoma tumor in mice, where we identified really great results, meaning that we can concentrate the amount of drugs that reach the tumor by injecting the MSCs loaded with these chemotherapeutic drugs. And as you can see in this mice, you can see that the drugs are really spread out inside the body of the animal. So meaning that our strategy is really prone and is really good to actually concentrate the drugs inside the tumor area. And in terms of reduction of the tumor, we achieved a really nice an effective shrinkage of the tumor after 35 days and after 4 different treatments. Of course, we have to improve this strategy a little bit more to be always more efficient and in order to be also translatable to humans. So what we are doing at the moment is to use a more physiologically relevant model. It's called orthotopic intratibial osteosarcoma model. So we do induce the tumor in the same position where normally the people or patients have the tumor. And in this case, we could prove that the retention of the MSCs, the combination of MSCs and drugs were intratumorally injected in the tibia is really efficient. And compared to the other delivery ways or compared to the drug by itself, we can avoid the spreading of the drugs and so the cytotoxicity and drugs. So again, we can achieve a double result, so concentration of the drugs and effective tumor reduction. We are in a phase of analyzing all the results, but at the same time, we are also in parallel conducting several studies in order to put us in the best condition to facilitate the path towards the clinical translation and have FDA approval for therapy. So what we do also for the cartilage project is to characterize the MSCs, meaning that we need to be sure that whatever we isolate from the different tissue, either fat pad or bone marrow, are real stem cells. So we have a way to do this characterization, and we always do them with our patients because every patient is different. So we might need to choose this therapy just for the best patients, meaning that the patient from which we can isolate the best MSCs population that we can. And the same for the assessor, the assessing of the biosafety profile of the MSCs. So we need to be sure that in the future, if we want to use the same stem cells affected patient, we need to be sure that these cells, they do not become cancer cells. So we have, again, a different test that we use in order to prove the biosafety profile. And we have already proved in a cohort of patients that these stem cells isolated from osteosarcoma or other bone tumor type cells are healthy and they are secure. So their biosafety profile is checked. The last thing that we do, and currently this diagram summarizes a technique that is currently under patent revision. So we want to find a way to isolate in the safest and fastest way the MSCs in order to achieve a so-called one-step surgical procedure. So we aim in the future to have a system through which the surgeon in theater will be able to isolate the stem cells and either use them to load with drugs to treat a bone sarcoma or to load them inside a chondrogenic hydrogel and repair the cartilage. And last but not least, I would like to acknowledge all the people that are working together in the cartilage regeneration team at the BioFab3D, and especially Professor Peter Chung and Professor Claudia Di Bella and Gordon Wallace, and all the PhD students and honor students involved, but also the funding agency that through those generous funds let us to continuously to investigate our research. And a second acknowledgement slide is for the osteosarcoma team, mostly based in Bologna, my hometown, where I started to work and with the NSCs, so the mesenchymal stem cells. And this final slide is to show you how the BioFab3D, so the biofabrication laboratory, will expand in the years to come, and it will become the ICONET Center for Medical Discovery, and to accelerate the collaboration in biomedical research to solve clinical challenges like cartilage regeneration or bone tumor treatment. And thanks to you.